God dag till ni alla och en varmt välkommen från en solig dag i Stockholm och Karolinska institutet. Jag befinner mig just nu i den här byggnaden, Aula Medica. Jag sitter längst upp här och det är lika solig dag idag som det ser ut på bilden. Jag har den glädje att få ekonomiskt stöd för mig alltid lite starkare. Och även att det är trist att man kan inte vara tillsammans med er face to face för liknande träff i år. Men jag är superglad att jag får den här möjligheten att berätta vad min forskningsgrupp håller på med. Vad vi åstadkommit under åren som vi har fått stöd, bland annat för mig alltid lite starkare. Jag tänkte att förklara att min grupp, jag är en preklinisk forskare. Jag jobbar tillsammans med kliniska forskare. Och tillsammans så hoppas jag verkligen att vi kommer lösa gåtan med ALS. Vi kommer förstå sjukdomsmekanismen och min fokus blir att försöka förstå hur vi kan rätta till vad det är som går fel. Så jag tänkte berätta 3 saker. Den första är en studie som vi publicerade förra året som visade vad vi kan åstadkomma. Fantastiskt intressant tycker jag. Och sen kommer jag berätta 2 projekt. En som ni har hört för mig förut och en som är ny vinkel på vad vi gör. Och jag hoppas att du får ursäkta att jag presenterar på engelska. Det går lite lättare för mig att prata forskning på mitt eget modersmålsspråk. So this study was published in the prestigious scientific journal Nature Medicine. Came out early on in the year. And the lead in this is my team leader, belongs to my research group, Sebastian Lewandowski. Everyone in red belongs to my group, and everyone in blue is a Karolinska researcher. Here you see Caroline, who will be talking to us today as well. And the other people are international colleagues, because this was an international study. And what Sebastian has done, he's interested in the vasculature, that means the blood vessels in the body. And he came up with this very interesting finding that a particular type of cell, which you find in the blood vessels called perivascular fibroblasts, the activity of these cells seems to be extremely important for how ALS disease develops. And I'll try and show you some data here and I'll try and explain it in a way hopefully that you will understand. What we use when we study ALS, we use experimental models, and there's a mouse called SOD1. So this is a mouse model of ALS, and what we're seeing here is the time course. And this is age in weeks of the mice, and what we can see is something happening with these graphs. So if we look here first on the right-hand side, then this side of the graph, this is showing the activity of cells, which we know in the brain become dysfunctional, and you can see that they come in a later point in the time course. It's here first at 16 weeks of age that the mice actually develop clinical signs of the ALS type disease, and it's when these cells are very active. These are the cells that I'm actually interested in studying myself. But what Sebastian could show was that before the symptom onset, there seemed to be activity of another type of cell which belonged to the vasculature, the blood vessels, which seemed to be very, very highly active at this early age. And when he studied, looked to see what these cells were producing, then this so-called perivascular fibroblasts seemed to be producing a protein, osteopontin, which we abbreviate to SPP1. If he then looked, he could stain. This is sections of the mouse spinal cord and brain, and you could see staining. And the red staining then is positive for this protein SPP1, and the blue is staining for another protein. That just shows you where you are. This is the blood vessel interior, this is the blood vessel wall. And here you can see in a control animal that's not sick that you have some type of coloration, the red here, but here you can see in the SOD mouse you have a much darker staining of the red, and that's telling you that this protein SPP1 is more highly expressed in the sick animal. When we were then able to look into human samples, both measuring the protein levels in the blood and also looking at sections from tissues from healthy people or ALS patients, then again this is showing the same blood vessel. You can see a slight coloration here, but in the ALS patient the staining is a lot more red, showing that there's more higher expression. And if you quantify this, you can see a very big difference in the expression of this protein in the patients. And when you look to see, we have a database that shows then what happens to the patients that are given these samples to our research. And this was done then with this international consortium. So it's samples from Holland, from Germany, from Belgium and Sweden, from Caroline. Then we can see that you could stratify patients into two groups. Ones which had low levels of this protein SPP1 and those that had high levels. And you can see that the disease development was a lot faster in those patients that had high levels of this protein marker in their blood. A big difference here. So what this study shows is it's very interesting because it says two things. It says one, that maybe measuring this SPP protein is the first biomarker to predict the disease course in patients with ALS. And secondly, we know where this cell is in the blood vessels in the brain, but it raises the question whether ALS starts as a dysfunction of the vasculature and not, as we previously thought, that it's a dysfunction of other cells which actually become activated a little bit later in the disease course. This study has received a lot of interest, and it's really nice to see Sebastian has been dealing with the interest that's come from society and from the scientific community. What needs to be done now is that other groups, other labs need to check to see whether this actually is a good, viable biomarker. Now we've shown the way that this is something that could be of interest to look at in the clinic, and we very much hope that the findings will be repeated by other researchers. But it's a landmark in the field because it shows that we potentially have the first biomarker for ALS, and we're very happy that we were able to achieve this. The second story then relates to the cells that I'm more interested in, and these are the guys that I think are responsible for actually the tissue damage. Even if they're not the first ones to start, they're the ones that we need to do something about if we want to have an effective therapy for the disease. So the cells I'm talking about are called macrophages, and they exist all over your body, and they have different names. So they're in the lung, they're in the liver, they're in your bone marrow, and in the brain these cells are called microglia. And this is how beautiful they are. You can see these green, they look like spiders almost with the long processes. The red is showing the blood vessels in the brain, in this tissue section. So they're very beautiful cells, and they sit there and they wave their arms and check to see what's going on. But if they misbehave, then we think that they can become very dangerous to neurons. And in this cartoon, then you see that these microglia cells have become red and nasty, and they're attacking the neurons and motor neurons in the case of ALS, and this will cause to then loss of neuronal function. So one thing that we'd like to achieve is to be able to get rid of these bad guys, and that's what you see in this thing. If we can go from this large number of cells that are misbehaving, and we can find efficient ways to deplete them, and this is what we've been developing during the past 2 years, and we now have a very good way of using a chemical which will do this. And then our hypothesis is that if we could repopulate the area, so we replace these angry red cells with these nice, calm, green-looking new versions of the microglia, then this would actually then enable the nerve cells to hopefully even be repaired, or at least for the disease process to be stopped in its tracks. So it's been quite difficult to work out the procedure for emptying the brain of all these cells and then repopulating it. But here we can see our first data to show these green dots here are cells that we've labeled with a fluorescent probe, and then in a mouse where we've depleted the microglia, we can then show that if we inject the cells, we can see them sitting here in quite nice numbers. So this gives us hope that we are actually able to achieve the first part of our therapy, which is then getting rid of the bad cells, putting new cells in. The other thing we've recently realized is that we can use another cell type, and this is called an amniotic epithelial cell. These are cells which are on the inside of the amniotic membrane around the developing placenta, and they can be harvested after birth from humans. And these cells are extremely immunosuppressive. That means they shut down bad immune responses. And what we use here is a model of multiple sclerosis. The reason we use this and not an ALS model is that this model takes 1 month from start to finish, as you can see here, 30 days, whereas the ALS model will take 1 year. So we can test the basis of the neuroinflammation in both models. We think is quite similar, so we can use this just as a test case. And what you can see in this graph is the disease severity, in this case incapacity of the animals. There was a paralysis that develops in this course, as you can see in the black lines. But if we treat these mice by injecting these amniotic epithelial cells that actually have an immunosuppressive effect themselves. Now, what this leads us to is that what we're thinking is that if we want to replace in ALS patients the dysfunctional microglia, we need to be able to give donor cells, but we can't take donor cells from the same person because genetically those cells are destined to misbehave. If we want to use human donor cells from healthy people, then there's a chance then if we transplant these that they'll be rejected. So now we can use these amniotic epithelial cells, which are extremely immunosuppressive, to protect the cells we're transplanting, and that's what we're showing here. So we worked out a protocol where we can make microglia-like cells from the blood cells of mice and man. So we don't need to harvest cells from the brain. We can actually start with something else and we can make them into microglia, and then we can harvest amniotic epithelial cells. And we then start with a depletion of the microglia, so we go from this side with a microglia dysfunctional to reduced numbers, and then we can inject our microglia-like cells, shown in the orange here, together with the amniotic epithelial cells. Then we think that this combined immunotherapy will lead to putting the CNS, the spinal cord, back into order and at least stop the disease in its track. So we're really, really excited by these results. It's been a lot of work and it's taken a lot of pain, but we finally got the pieces of the puzzle together. And in the next phase, in the next year, what we'll be doing is we'll actually be testing this in our models and working out the best way. We need to work out how to get maximal numbers of cells into the mouse in order for this to have an effect. But we're really, really excited that we've come this far in quite a short time. The next story, the last story, is an ongoing project, which is also another way to approach these cells, these microglia that we think are dysfunctional. And again, I'm going to show you some data that we've used in another inflammatory model, but we're just ready to go, and I hope you get excited by this as I am. So one of the challenges of trying to find new drugs, to develop new drugs and new therapies for diseases, is it takes a lot of time and a lot of money. To identify a new drug and bring it to market is usually 20 years of work and the tens of millions or hundreds of millions of crowns. So I have a very smart PhD student, Kaying Zhu, that came up with this idea that why not look into the drugs which already exist, so-called FDA-approved drugs, so they're already being used. And can we identify a drug that will have the properties that we want to do? And the properties that we want to do is then if we take our microglia, can we modify their functionality? And that's what this scheme is supposed to show you. So what he's done is be able to go into a database where other people have done this and deposited their data, and they've taken these cells and they've acted them in different ways, and they've looked to see what happens to them, and they've looked to see what gene profiles they express, and they've come up with an idea about what certain drugs will do. And we identified this drug, topotecan, which is used today for treatment of ovarian and lung cancer, and it seemed to have an effect on microglia in the way we'd like it to be affected. So what we then did is to be able to take this drug and to test it in our neuroinflammation model, our MS model again. And here again we have disease course with time, and you can see that the untreated animals get disease as usual. But if we then start the treatment using this topotecan drug in these animals after the disease has started, then we can significantly reduce it. So this is showing that we're having a good immunomodulatory effect, and we know that this drug is having an effect on the cells we're interested in, the microglia. Now, the other next challenge is, well, how will we make sure that this drug is only delivered to the cells we want to do? And now we come into a really cool technology, which is called DNA origami. So I work with another good colleague, Björn Höberg, and he works with these DNA molecules. And what one can do with DNA is one can make them into different types of structures, and he makes them into this rod-like structure. And what we asked him was, well, can we add on molecules onto the surface of this DNA construct? And one of them is a protein called glucan, which comes from yeast, which we know then will interact with the receptor, which is expressed on these microglial cells that we're interested in. So by adding on the glucan onto the DNA origami, we can make sure that they only get delivered to cells which express this receptor, in our case, the microglia. And then we can package our topotecan drug inside this and see what happens when we put them into mice. And that's what you see on this graph. So what we see is the same thing again, the paralysis with time, and here is the ones with this super construct of the drug in the DNA origami, which is even doing better than we gave the free drug itself, because we're delivering it to the cells we want to do. And this is an index showing disease burden in these mice, if they're untreated, if they're treated with a control, or if they're treated with this. So we're really excited by this prospect, and we think that this will be a smart way to have a delivery to the cells we're interested in. And now it's just tested in our ALS models. Så det var allt som jag tänkte berätta. Jag hoppas att kunna följa med. Jag tycker att det är intressant för oss att lära oss steg för steg vad vi kan göra. Och det blir en tyvärr långsam process, men det är viktigt att det går till på rätt sättet. Men min ambition är att tillsammans vi kommer kunna göra någonting, make a difference, bli mad, det är vad jag brukar säga. Och kom ihåg att jag har sagt, jag har min alltid lite starkare armband. Jag har det nu bredvid min klocka. Jag påminner mig varje dag när jag tittar på den att klockan tickar. Och det är dags att vi gör någonting, men vi jobbar så gott som vi kan. Och jag hoppas att nästa år så har jag ännu mer glada nyheter för det. Tills dess, sköt om alla.